This article, titled A Data-Driven Approach for Soft Tissue Biomarker Identification Linked to Chiari-like Malformation and Syringomyelia, published in the journal Frontiers in Veterinary Science on January 22, 2025, explores the use of machine learning to identify morphological features associated with Chiari-like malformation, CM, and Syringomyelia, SM, in dogs. The study uses magnetic resonance imaging, MRI, scans and a data-driven approach to find biomarkers for these conditions, aiming to improve diagnosis and identify at-risk dogs. Introduction. Canine Chiari-like malformation is a complex developmental condition characterized by a conformational change of the skull and craniocervical vertebrae, leading to overcrowding of the brain and spinal cord. This overcrowding is often due to skull base shortening, craniofacial hypoplasia, and abnormalities at the craniocervical junction. Brachycephalic toy dog breeds are particularly susceptible to CM. A significant percentage of these dogs, even those without clinical signs, show some degree of craniocervical morphologic abnormality. Dogs with more extreme brachycephaly can show signs of pain, known as Chiari-associated pain, CMP, which includes vocalization on rapid postural changes, signs suggesting head or facial pain, reduced activity, spinal pain, and behavioral changes. These signs can drastically reduce a dog's quality of life. Additionally, morphological changes linked to CM can disrupt the flow of cerebrospinal fluid, CSF, leading to fluid-filled cavities in the spinal cord, known as Syringomyelia, SM. These cavities, or syrinxes, can cause pain, including allodynia, a maladaptive scratch reflex, weakness, and other neurological signs. Brachycephalic toy dogs are at higher risk of developing both CMP and SM, and these conditions are often studied together. Both CM and SM are heritable conditions, which can worsen over generations, emphasizing the importance of early detection for breeding choices. Currently, diagnosis of CMP and SM relies on magnetic resonance imaging (MRI). While SM can be identified and measured objectively, diagnosis of CMP is more subjective, relying on a combination of clinical signs, MRI, and the elimination of other causes of pain. Prior research has used statistical analysis, morphometric measurements, and machine learning to explore the morphologies linked to both conditions, with machine learning showing promise in automating the diagnostic process and identifying subtle patterns that may be missed by human observation. Study design. This study aimed to extend previous research using machine learning to identify novel discriminative regions associated with CMP and SM, using a data-driven approach across the entire sagittal image of the canine head. The study used mid-sagittal T2-weighted MRI slices from 120 Cavalier King Charles Spaniels (CKCS), a breed known to be predisposed to these conditions. The dogs were categorized into 3 groups based on physical exams, medical history, and MRI findings: CMN, clinically normal, CMP, CM with associated pain, and SMS, CM with Syringomyelia. The CMN group consisted of dogs at least 3 years old with no history or signs of CMP or Syringomyelia. The CMP group included dogs with CM identified on MRI and clinical signs of CM pain, with a central canal diameter less than 1 mm. The SMS group included dogs with CM and clinical signs of SM, with a syrinx maximum transverse diameter greater than 4 mm. Dogs with smaller syrinxes were excluded from the study. The study was approved by the University of Surrey NASPA Ethics Committee. The methodology had 4 key steps, summarized in Figure 1: image preprocessing, feature selection, feature evaluation, and inference of morphologies. The overall process used machine learning to explore morphological trends linked to these disorders. All experimental processes used MATLAB software. Image preprocessing involved selecting a reference dog from the CMN group. This reference was used as a template to warp the MRI images of all other dogs using image registration. The warping process created maps of pixel displacements, which were then analyzed to identify local candidate regions indicative of CMP or SMS. Feature evaluation used machine learning to identify the most predictive regions, which were refined to enhance predictive performance. The study selected a reference dog by manually annotating key anatomical landmarks on the MRI scans of all 34 CMN dogs and analyzing these using Principal Components Analysis, PCA. Figure 2 illustrates the 7 anatomical structures used in the analysis, including the center of the intrathalamic adhesion, the mammillary body, the apex of the fourth ventricle, the tip of the caudal cerebellar vermis, the occipital pole, the center of the CSF space between the frontal lobe and olfactory bulb, and the junction between the hard and soft palate. The chosen reference dog's age and weight were representative of the CMN group. After selecting the reference dog, all subject images were registered to it, first using a rigid body registration to align the images, followed by non-rigid Demons registration, which is illustrated in Figure 3. Demons registration applied nonlinear transformations to align the query image to the reference image, creating a deformation map showing pixel displacements. The feature selection step involved examining each pixel and its local neighborhood, referring to them as seed points, to find potential biomarkers. A 9 by 9 pixel region around each seed point was selected based on pilot studies and is shown in Figure 4. These local displacement maps were then used to build machine-learned features to predict CMP and SMS. In the feature evaluation step, a support vector machine (SVM) algorithm was used to evaluate local displacement maps. CM and SM experiments were carried out separately, with the CM experiment treating CMN as controls and CMP as disease subjects, while the SM experiment treated CMN as controls and SMS as disease subjects. Each local displacement map, identified by its central pixel, was evaluated using receiver operating characteristic (ROC) curves, and the area under the curve (AUC) score was calculated, with scores approaching 1.0 indicating perfect predictive performance. The datasets were divided into training and testing sets, with a ratio of approximately 80/20. The training dataset was grouped into 8 folds for cross-validation to see how well the predictive models performed. The average performance across folds was visually represented by mapping pixel performance across the reference image. The map of AUC performance was threshold at 0.62, and the regions were repeatedly dilated to maximize performance. Finally, an exhaustive search of all possible combinations of these regions was conducted to find the combination yielding the highest AUC score during training. Results for CMP: the study identified 4 key regions as highly predictive, shown in Figure 7a. Surprisingly, all 4 were outside the brain, with 3 outside the nervous system, the caudal end of the soft palate, the larynx and soft tissue dorsal to the larynx, the fascia containing the carotid artery, and the level of the C3 spinal cord segment. The machine learning model using the combined morphological data from these 4 regions yielded an AUC score of 0.87 with 89% sensitivity and 76% specificity. Figure 7b shows the corresponding ROC curve. PCA analysis was used to understand morphological changes in the 4 regions, shown in Figure 7c. This showed that the soft palate exhibited ventral displacements, the larynx showed caudally and ventrally directed morphologies, the carotid artery area showed opposing morphologies, and the C2/C3 spinal cord showed different directions of displacement. Figure 7c shows the average displacements and directions for CMN subjects and deformations linked to CMP. For SMS analysis, the study found more regions with AUC scores above 0.62 compared to the CMP study, as shown in Figure 8 and Figure 9. After optimization, the study identified 14 strongly predictive candidate regions associated with SMS. The best combination included 4 regions: the rostral cerebellum and tentorium cerebelli, the brain stem ventral to the fourth ventricle, rostral to the soft palate, and the epiglottal region of the larynx, shown in Figure 10a. The corresponding ROC curve showed an AUC score of 0.85, with 84% sensitivity and 80% specificity, as shown in Figure 10b. The model correctly classified 76% of the CMP dogs as not being SMS. PCA analysis of these regions, shown in Figure 10c, revealed distinct morphological behaviors, with dorsal morphologies for SMS at the tentorium cerebelli and brain stem, differing from the ventral morphologies in the CMN dogs. The soft palate and larynx regions showed different angles of separation between CMN and SMS subjects. Figure 10c shows the directions of deformations linked to SMS compared to CMN subjects. Discussion: the study used a data-driven approach to identify soft tissue deformations predictive of CMP and SMS. The consistency of a single neurologist assessing the MRI data minimized interoperator variance. The machine learning algorithm identified regions of interest and optimize the set of regions linked to disease without any prior knowledge. The study achieved up to 89% accuracy for CMP and 80% accuracy for SM. The study found that all major predictive regions for CMP were surprisingly outside the nervous system. The conformational changes in the skull may also cause changes in the upper airway and associated soft tissue. Alternatively, changes in the airway may be affecting intrathoracic pressure and sleep, which may affect cerebrospinal fluid movement and clinical signs. Also, changes in the great blood vessels might affect drainage from the head. The C2/C3 region, which is sometimes curved, was highlighted in the study for CMP. Figure 11 shows an example MRI scan of a dog with CMP. For SM, two regions outside the central nervous system were identified, similar to the CMP results. The location of these regions close to the soft palate and larynx could be due to a shared disease pathogenesis or para phenomenon relating to the Valsalva maneuver. The rostral cerebellum region, a part of the brain which is often displaced in CM, and the brain stem region were also identified as predictive for SM. The study noted that the morphological changes in the study may relate to these known clinical changes. The study also showed that the findings generalize effectively across the wider population using a bootstrap dataset. The limitations of the study included the use of a support vector machine, which may be less optimal for larger datasets, and it also depended on a representative reference dog. Also, the initial rigid body registration before the demon's registration may obscure morphologies. The study also noted that many veterinary MRI images are not of a high standard, which machine learning tools must be able to overcome. Additionally, the current health schemes do not always require a full brain MRI, which can limit historical image analysis. Future research would benefit from a larger dataset and a tool adaptable to variations in imaging. Further investigation will extend to three-dimensional morphologies. The same methodology could be developed for other brachycephalic breeds and conditions, such as brachycephalic obstructive airway syndrome, BOAS. The study's results suggest the methodology could successfully analyze morphological changes due to developmental disorders without prior biological knowledge. The study identified internal morphologies of CKCS subject to CMP and SMS, distinguishing between disease-related deformations and normal variations. The results serve as a proof of concept for using machine learning for diagnostic medical imaging in CMP and SMS, representing state-of-the-art performance. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.